the nucleotide based and another is the protein based, and both were having the different types like primary and secondary. And we have discussed about the examples like a gene bank, EMBL, DDBJ, and others. Then after that, we have gone for the ORF finders to how to find a, like if you are having a genome sequence, a gene sequence, then how to find a ORF from there, open reading frame from there, because open reading frame is only going to be translated. After that, getting the ORF region. So if you remember, we have taken a gene that is the AY221982, and we checked the length. So total length was the 972 base pairs. So when we have used this 972 base pair and we checked in the ORF finder, we got different size of the ORF, and among them we have picked up the ORF which is having the 807 base pairs, and that 807 base pair is going to give you the size of the 268 amino acids. So those 268 amino acids, if you remember, then I took you to show some different kind softwares like a protparam to know what will be the molecular weight of your protein, what will be the pi of the protein, how many cysteine residues it will have, what will be the extinction coefficient, and etc. After knowing all those things, if you remember, the molecular weight was around 30 kd, 29 point something. pi was around 5. It was acidic protein, and then then it doesn't contain any cysteine residues. So then after that, we have explored the another software that was the signal P. So signal P we have used to find the signal peptide in the protein, because mostly proteins when they translate as a precursor protein. So precursor protein may have the signal peptide. To know whether it is having signal peptide or not, we have checked the signal P server, and there we found our protein is having a signal peptide which is having the cleavage site between the 23 to 24 amino acids. After knowing this, that yes, our protein is having its cleavage site, we have tested another server, that was the TopCon, to know whether our protein is having any signal peptide or not. Sorry, transmembrane domain or not. So after analysis, we got the information that yes, it is having the first 22 amino acids are forming a transmembrane region. So after knowing that, now we have the information that yes, our protein is having a signal peptide also, and it is having the transmembrane region also. If you see, both are having one common thing, that is the hydrophobicity of the region. So both are, if you see, the position is also same. So if you want to clone this gene of interest, or if you want to express the protein, what we have to do? We have to express the protein or after removing this 23 amino acids, because these 23 amino acids are containing the single peptide, and these 23 amino acids are also having a transmembrane domain. So these things will interfere in the purification. So I hope all of you remember these things. Then after that, I took you to the expression vector. Why we need expression vector, I have explained you, because there is two type of the vector which carry generally DNA molecule, and that they help to express the DNA molecule, and later they help in the purification. So the vector we have discussed, generally there is two type. One is the cloning vector and another thing is the expression vector. Yeah, you can please stop me if you are having any doubt. Okay, so cloning vector actually, which is just used only for the simple cloning. Okay, like I want to clone a gene, I have taken this vector, I have inserted, and I have multiplied. That's all. But if I want to express the protein, we have to have a very specific vector. A specific vector means which sense? It should have the complete transcription machinery, because for the transcription you need a promoter, you need a operator, you need a RBS, ribosome binding site. You should have the initiation codon, you should have the termination codon. So to have all these things, it is difficult to get in a cloning vector, because cloning vector is only just for the amplification, to amplify or to increase the number of the particular gene of interest, but expression vector to express the protein. So if you remember, I have shown you one example that was the PET28 vector.
सर आप स्लाइड चेंज कर रहे हैं कि वही स्लाइड पे है?
No, I am not changing, because just I am taking them, I am refreshing them. If you want, okay.
Okay.
I will change now slide actually.
ठीक है, ठीक है.
Yeah. So I have discussed with you about the this PET28 vector. So now today, if you remember, yeah, one more thing we have discussed, that is the how to design the primer. So if you remember, when we were designing the primer, I have shown you there is two ways. One is the just you take your sequence, put in the server and say these are the parameters which I am looking. So if you remember, when I was discussing about the parameters, so there were several parameters. First was the length actually. So length means what should be the length of your primer. So if you remember, the length should be around 18 to 30 30 base pairs. Okay, I will take you here to previous class actually. So length should be 18 to 30 nucleotides. TM should be 55 to 65. And the difference between the two primers, forward and reverse primer, not should be more than 5 degrees centigrade. If your TM is not good, you should look for the GC content. GC content should be between the 40 to 60 degree, 60%. Another thing, you should not have the complementation with each other, both forward and reverse primer. And very important thing, you should have the C or G at the 3' of the primer. So these things I have discussed with you. Then I have shown you the one expression vector PET28. So if we want to clone our gene of interest, which we have got, that length of the gene of interest is the 807 base pair. If we want to clone, so for cloning you need an expression vector, and expression vector contains the MCS. MCS called multiple cloning site. So if you try to recollect, you can see here multiple cloning site. Okay, so this multiple cloning site is having various enzyme, BAMH1, EcoR1, SEC1, SAL1, HINT3, NOT1 and XHO1. So we have to find the enzyme which are compatible for our insert. For that, what we have to do, we have to run a program that called the anybody remember what program I have shown you that day? Sir, can you please unmute these people so we can have discussion kind things? Okay, so just I will tell you, because that enzyme, that software which we have performed that day is the NEV cutter. So I will run today again to show you which, because if you remember, now your restriction, your vector is having limited restriction sites. So what we have to do, we have to look the restriction site which is compatible to your expression vector. Okay, so this is the 807 base pair, and now I have clicked here. I hope all of you are able to see. Okay, so that day also I mentioned you, like in this you can get a list actually that how many enzymes are able to cut at one place means one cutter. How many enzymes are able to cut at two places? This restriction, this DNA or this gene actually, then it is two cutter, and if they are able to cut more, then according to that you can see. But we are not interested in how many places they are able to cut. We are interested in an enzyme which should be compatible with the our expression vector. So we have to look at zero cutter. Which enzyme is able to cut only zero time to this gene? So if you remember here, you are having here BAMH1, you are having EcoR1, you are having SAL1, you are having HIND3, you are having XHO1. Okay, I am not taking the not one actually, because the compatibility generally creates problem with the not one. Okay, so I have taken 6 enzymes, 5 enzymes from here, and now let me check how many enzymes are not able to cut our gene of interest. So let's look alphabetically. So first is the BAMH1. So BAMH1 is able to cut. That is the reason it is not part of the zero cutter. If it is part of zero cutter, that means it is not able to cut. So if you see in this list, you are not able to see the BAMH1. So that means BAMH1 is ruled out. You cannot use BAMH1 for cloning of your gene of interest. Then next one is the EcoR1. Let's check the EcoR1. I hope all of you are able to see the screen.
Yes, sir. We are able to see the screen, sir.
Thank you. Okay, so then you see now EcoR1 is there in the list actually. So what is the meaning of this? That means EcoR1 is not able to cut this gene. So that is mean the gene of this EcoR1 is significant for us. Then let's go now check the next alphabetically, HIND3. So you see HIND3 is there, is not in the, okay, it is there on the 92 position. If you see, HIND3 is there. So HIND3 means HIND3 is also not able to cut your gene of interest. Okay, so you are going to consider this one. Okay, then next one is the SAL1. So this see, SAL1 is also not there in the is there in the list. That means it is also unable to cut your gene of interest. And then let's check the XHO1. Yes, XHO1 is also there. Okay, so now you come back to the vector. So among five, 4 is unable to cut the gene of interest. So that is good thing for us, because we are having now a lot of options. If suppose if you are having only 1 or 2 options, then you are in the trouble, but you are having many options here. So like let's target. So according to you, which one will be the more suitable enzyme for the cloning here? Can you please tell me? Okay, so generally we see the compatibility of these enzymes. So because these enzymes mostly are compatible with each other, either you buy from the NAV or you buy from the fermentase. So they are generally compatible. Now next thing we have to see the distance among them. See, if you take the EcoR1 and BAMH1, there is very small gap actually. And all of you know, when any restriction enzymes cut, firstly it does recognition and then it does the restriction. So if enzyme has to work, firstly it has to recognize. Now suppose what happens if you take both BAMH1 and EcoR1. BAMH1 restriction site is up to here and then EcoR1 is starting. So suppose BAMH1 has gone and it has cut it. Now it will face the EcoR1 will face the problem to recognize the restriction site, because there is not any additional base pair. So we have to look always enzyme which are quite far from each other. So now here you see EcoR1 is here and HIND3 is also far, XHO1 is also far, SAL1 is a little bit closer. So what we will do, we will take the EcoR1 and we will take another enzyme XHO1 because it is far. Okay. So after getting these information, now we have selected two enzymes. One is the EcoR1 and another is XHO1. So as well, if you know that we have to design two primer. One is the forward primer and another is the reverse primer. So how we will design the forward and reverse primer? So if you want to design the forward and reverse primer, I will just take you a little bit on this board actually. I hope all of you are able to see the board. So here, this is your gene of interest. Okay. Are you able to see or not?
सर बोर्ड तो दिखाई दे रहा है लेकिन बोर्ड पे जो लिख रहे हैं आप वो नहीं दिखाई दे रहा.
Okay, okay. I will take it closer. The camera. Okay, just I have draw one line actually. That line belongs from the our gene of interest. I hope now it is visible.
हल्का हल्का दिखाई दे रहा है.
Okay. Is it, sir? Now okay? Okay. So now this gene of interest, just after doing this exercise, we got the information. This gene is not having the EcoR1 and XHO1 restriction site. So what we have to do? We have to design one primer from here. That will be our forward primer. Another primer we have to design from here. That will be our reverse primer. So what we will do now? Because this EcoR1 and XHO1 is not there in your gene of interest. So what we will do? We will add those EcoR1 and XHO1 as an adapter in our primer. Okay, so primer, forward primer means it should be complementary of your first strand and means positive strand, and reverse primer means it should be complementary of your negative or anti-sense strand. Okay, so here now we have to add the restriction site. Okay, here suppose add the EcoR1 and here you add the XHO1. Because when you will add this EcoR1 in the forward primer and in the reverse primer, so when this will amplify the gene of interest, this EcoR1 and XHO1 will come as an overhang in your gene of interest. So then your gene of interest size will increase from from up to, like you see, your gene of interest is starting basically from ATG. Now just before ATG, you have to add the restriction sites of the EcoR1. Okay. And similar way, you have to add the restriction site for the XHO1 at the C prime. XHO1 C restriction site is CTC GAG. If you see in this, generally what happens actually, when you want to express a protein, during expression there is a problem. What happens? Because if you see, firstly you are getting histidine tag, then you are getting the ND1, NHE1, T7 tag, and VMH1. So this many amino acids comes additional in your gene of interest. To ignore that, what we do actually, generally we take ND1 as in the forward primer and XHO1 in the reverse primer. So now you see, you have added the here EcoR1 and XHO1 for this. Now what will happen if your gene of interest is extended? It is up to from here to from here. Okay. So what will happen? Now when you will cut with the EcoR1, you will get easily overhang, and that overhang, similar overhang will be present in your expression vector. So that is the reason you have to introduce a restriction site. But what happens, just now I mentioned, always enzymes works in a restriction enzymes work in two ways. First thing is the recognition and second thing is the restriction. So now what will happen, because it is starting with the EcoR1 only. So then this is not a good thing to start with directly with the EcoR1. So what we have to do, we have to add some additional base pair before the EcoR1. So then now suppose generally we add the three base pair. So one is the A, T. You can add, it is your call actually. You can add C, G, C, something which should not be cutted by any other engine. Okay. So then A, T, and then you have the eco-carbon, and then you will have the engine for the, sorry, your gene of interest. Similar way, you have to add here you have to add before restriction site. And in the reverse primer, you have to add after the reverse primer. Okay. Sorry, after the restriction site. So this is the these are the two things which you have to take care when you are designing a primer. Now, how to check the different parameters for a primer? Because that day I saw, if you remember, I saw you one software, the one server that is the primer 3 that calculate everything for you. Okay, you need not to worry about the thing. Okay, but here, because you are designing manually, so manually when you are designing, you have to be more careful. So let's design by our own actually. So I just mentioned length should be from 18 to 30 amino acids. So I hope all of you are able to see this sequence of the ORF region. Okay, so it is starting with the ATG. So let's take firstly around 20 amino acids, 20 base pair, 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 16, 17, 18, 19, 20. Now here problem actually. If we take these 20, what is the problem? If you will take these 20, the problem will come with the thumb rule of the primer designing. According to primer designing, you should have either G or C at the end of the primer, like means at the 3 prime. So now you see, you have taken 20 base pair from this sequence to design the primer, but it is ending with the T. So that is not a good choice. So what we have to do? Either you have to reduce the size of the, because if you see on 18th position, you are having the G. But 19 and 20 both are A and T. So both are not good. So either you reduce the size or you increase the size of the primer. Reducing size is not suggestible in this case. Why? Because this gene you are going to amplify from a chickpea. And chickpea is a plant, and plant is having many millions gene actually. So if you are having a small primer, then getting non-specific amplification is very common. So that is the reason what we have to do. We cannot reduce the size of the primer, because when you will reduce the size of the primer, non-specificity chances will be increased. So now what we have to do, we will increase the size. So now see, after this 80, then there is another 80. Then next one is the T, that is also not working for us. Then another is also T, and then after that there is a C. So that's mean due to this technical issue, you have to add the additional 5 base pair in your sequence. Okay, now you got this. Now just now I mentioned you that you have to take care of the, now after before this, because this is your forward primer. So now you have to design the reverse primer. So if you want to design the reverse primer, you have to add the restriction site of the eco-R1. Eco-R1 restriction site is the GAA-TTC. Okay, so let's add the GAA-TTC. GAA-TTC. Okay, so now you are having the restriction side of the eco-R1 also. So when eco-R1 will cut, it will cut here between the GA and now the length has reached to the 25 was there earlier, and now 6 is 6 has come from the restriction side, so it is the 31. I told you you need something for for the recognition, because this is the recognition side. Between G and A, it will cut actually. So you have to add the some additional nucleotide. You can add like A, A, T. Now after adding these things, now next step will be to check the, now if I ask you what is the TM, anybody can tell me the what is the TM? TM you can do after calculating by the that simple formula 4 into G plus 4 G plus C plus 2 A plus T. You can do by that way, but you may not get precise. So to get the precise thing, what we have to do, there is one server actually that called the OligoCalc. Okay, I hope all of you are able to follow me. So let's go to OligoCalc. Okay, this is a server which will give you the all parameter of this nucleotide, this primer, which oligonucleotide, which you have just designed. Okay, so take this one. Copy your primer sequence which you have designed and paste here. Okay, and there is one down, if you see, there is one option to calculate. Now just I press this calculate, I got two things. One is the TM. TM is the 56 degrees centigrade. Another is the GC content. GC content is just only 26%. So this is very less actually. Even TM is also very less and GC content is also less. So in that case, what you have to do? You have to improve, you have to increase the length further, because it is not giving you, because if you are having the TM 56 degrees centigrade, you are going to perform your PCR at the 50 degrees centigrade. And at the 50 degrees centigrade, you are having very high chances to get the non-specific amplification. So that is the reason it is better to take the some more nucleotide before proceeding with this. Okay, let's see now. There is, I have taken another 4 because 25th is the C, but 26th is the T. So then it is not going to work for us. 27 is A, that is also not going to work. 28 is the T, then that is also not going to work. So then next one is the 29 is the C. So let's add those nucleotide also here and then calculate the TM again by the using OligoCalc. Go here, remove, just clean this one and paste the new sequence and calculate. See, it has reached to the 58 degrees centigrade. It is increasing, but it is not increasing as per our expectation. GC content is also is almost constant. So just when I was taking this sequence, I realized there is, after this C, there is another C actually. So let's take that C also in the consideration and then calculate. Wow. So it has reached to around the 59.2. So that's okay for us, and GC content has also reached, because now we can do around 54 or 55. We can perform the PCR. So that is not a bad choice. Okay, and that length has also increased, so non-specific chances are lesser. And another thing I will not add actually, because mostly companies, when you are going for synthesis of this primer, they charge for the 10 molar, 10 nanomolar. They charge, sorry, for up to 39, they take the 10 nanomolar. So then charges will be lesser. If you will increase further, then you have to pay double charges. So that is the reason I feel this is fine for me as a forward primer. Okay, so it is containing the both things. It is containing the first 3 is the your additional base pairs. Then this 3, then next 6 is the from the eco-carbon restriction site, and then this is from your gene of interest. And if you see, like this is now all together, you are having the 30 base pair from your gene of interest. So this is fine for me. I hope this it is fine for you also. So I will take this one as a my forward primer. Okay, so this is going to be my forward primer. Okay, I will write down the all prime, this thing also, what is it? Uh, Parameters also, because the parameters should be identical for the reverse primer. So parameter just now we saw for forward primer, what is the TM? TM is the 59 degrees centigrade. Okay. So now when we are designing the reverse primer, that time we have to take care of this thing. The TM should not be more than 5. So what we will do now, we will take the reverse primer. Reverse primer, how you will get? You take again 20 or 22 base pair from the reverse from the 3 prime of the your uh, uh, gene of interest, and then go here, and after putting here, what you have to do, you have to you have to swap the strand. Okay, why you have to swap? Because this is going to anneal with your strand. Okay, so then it should be the reverse of this. So that option you can see here, swap strand. Click on swap strand. Now it has gone up. Okay, further if you then it is back. And if you remember, I told you you have to add the restriction site for the Jo1. So let's add the restriction site for Jo1. CTCGAG. So this is the restriction site for the Jo1, and as I mentioned you have to add the additional 3 nucleotides. So let's add same AAT and then calculate. You have calculated and it is coming around 61.4. So, but you don't need this one as such. You need the swap strand. So just you swap the strand and now you check the parameter. The parameter will be same. It is around 61.4. Length is the 28. So if you see here, the problem is the length actually. Why length is the problem? Because you are having the 28 base pair, and in 28, 6 has come from the restriction sites, 3 has come from the additional base pairs. So 9 is basically additional amino acid base pair, while you are having only 19 nucleotides from the your gene of interest. So then it is not a good thing. So what we have to do, in that case we have to take some more additional nucleotide from the our gene of interest. So let's take, now the problem will come actually if you go and try to take from there. See, another one is the, after G it is the A, then T, and then C. So then now that's means you have to take not one nucleotide, you have to take basically 4 nucleotides to make the last one as a C or G. Okay. So let's copy again in this. Okay, so let's, I have calculated, and if you see, now it has reached to the 63.1, but still, because your forward primer is in 50, is 59 and reverse is 63, so still it is in the range of the 5 degrees centigrade. So then there is not any problem. You can consider this one because it is having the 32, so length is also enough good actually for the amplification, and it will not have the chances to get the non-specific amplification. So this one is going to be our reverse primer. So what will happen when you will amplify? Now your gene will have the extension of this thing. They will have the additional nucleotide along with the restriction sites. So this adapter region you will get, and TM is almost 69, 63, so you can perform 55 around PCR. So this is a way to design the manually primers. If any question is there, please ask me. Sir, any question is there? Please ask me.
Participants, please, you can unmute yourself. I have made the option of unmute. If not, you can put in chat box at least. As of now, no questions are found, sir. You can continue.
Okay. Okay. So now this way, we will get our construct, and construct will have the pad 20. Construct will have the pad 28A, and pad 28 is having all machinery for the transcription. Now you take this pad 28A, put in the pad 28A construct with the your gene of interest. Now put in a expression host. So what are the expression hosts? So generally we use bacteria as a expression host. Now you will ask me, this is a plant protein, why you want to express in a bacterial system? Why you don't want to use another system? Why I want to use this expression system as a bacteria? Because bacteria is having a lot of benefits. First is it is very cheap actually to grow the bacteria, because for bacteria you need only the LV broth. If you are having E. coli, just you need the LV broth, and LV broth needs only three components. One is the yeast extract, another is tryptophan, and third one is the NaCl, and NaCl is easily available. Yeast extract and tryptophan is also not that much expensive. First thing is the cost. You are going to spend very less cost. Second thing is the time, because if you see any other expression host, either you see yeast, or you see human or mammalian cell, or you see the insect cell, all these hosts are needs lot of time, while E. coli is having very less generation time. That is the just 20 minutes for a generation time for E. coli. So if you will grow overnight, by next morning when you will come, you will have the plenty cells, and in those E. coli cells, because you are having your gene of interest, your construct there, so when E. coli will grow, your protein will also express there. So first thing is the cost, second thing is the time, and third another thing important is the yield, because generation time is less, so overnight culture will give you enough number of cells, and and all cells containing your gene of interest, so then they are going to express the particular protein also. So you will get a plenty protein from the E. coli. Now let's go for the other system. You may ask why we are not using the yeast, and why only E. coli? Is E. coli a universal system to grow the any gene of interest? No, because E. coli biggest problem is the post-translation modification. Because E. coli is a prokaryotic system, and you are having a eukaryotic protein. And this eukaryotic protein, just now, if you remember, first day also I mentioned the name, and even in the slide you can see the name of this protein is the lectin. And lectin is containing the carbohydrate binding moiety. Okay, so then this needs the post-translation modification. That means it wants to go for it needs the carbohydrate moiety, so that means it contains it should go for the glycosides. Then for this, E. coli is not a good system. For this, you have to look a cell which can perform the post-translation modification process. But the most common and standard system is the E. coli. Like in the 99.9% cases in my lab, we use the E. coli. Then next system is the yeast system. Why people prefer the yeast? Yeast is cost-wise is not very high, but generation time of the yeast is lesser than the E. coli. That is the reason, E. coli you just grow overnight. But in the case of yeast, you have to put 2 days, same culture, to grow. So then you have to spend additional time. Another thing is the yield. Yield will be very good actually, but not equivalent to the E. coli, because E. coli has grown up in the night itself. So that is the reason. But yeah, east you will get some kind post-translational modification. Third system is the insect system. That is very expensive system. You need a different setup. You need everything different, and yield is also not very great. Cost is also high because you need specific cell lines to grow them. The but cheapest, but a very good thing is the you will get the post-translational modification because that is a eukaryotic system. Similar kind mammalian system, that is also eukaryotic system. So you will get a post-translational modification. Only the problem will come when you are looking for the cost and other factors. That time you will face the problem. So that is the reason most preferred system is the E. coli. Now you have grown up your protein in the E. coli and you got the plenty number and you want to go further, and that is for the chromatography. But before going that, actually you can check the other parameters of this protein also. There is one more parameter that called the protein family. Like here, because this is annotated. Suppose you in the other case you got a gene of interest that is not having any kind of annotation. So what you can do? One thing you can perform the sequence alignment. By sequence alignment, you will get okay who is the similar or identical gene of interest available in the database. So then my gene of interest or my protein will be the identical. It will belong maybe from the same family. Or in another case, what you can do, you can run a server. That server called the pfarm. Please try to run parallelly whatever I am doing actually. So go to pfarm, click here pfarm, and then you now copy the, because in pfarm you need the your protein sequence. So protein sequence length is the 268 amino acids. You copy these 268 amino acids. It will not have 269 because all of you are aware, because that one amino acid is a termination codon actually. So then you will not get that one. Okay, let's paste 268 amino acids here. There are several options: quick links, sequence search, view a pfarm entry, view a claim, view sequence, view a structure, keyword search, jump to. Okay, so we will go to the sequence search. Let's paste your sequence here and then click on go. Okay, let it run. It will take some time. Okay, so result has come here. So as you know, in beginning itself, we know from beginning itself that this protein is a lectin. So you see here in this first thing is the diagrammatic representation, which is mentioning this is having a region 26 to 267 amino acids. Total, how many you are having? You are having 268. It is saying from 26 to 267, it is having the it belongs from the lectin legume B family. If you want to read leg means legume B means Leguminaceae B family. So all of us know because this is protein from the chickpea, and chickpea is a member of the Leguminaceae family. So then definitely it is matching whatever knowledge we are having. But because this is a non-protein, so you are having all that knowledge. But if you are having unknown protein, then what you will do? Another thing, it is telling you one more important thing, that only it is that lectin domain is starting from the 26 amino acid, not from the first amino acid. So if you feel that I just want only the lectin region or lectin domain, then you can trim this region also when you are going to amplify. But generally, when you are not having similar peptide or transmembrane region, no logic to remove, and just 26 amino acids, they are not going to hurt your protein. So let it be them there. Okay. So then it is saying you this belongs from the lectin leg family. Another is that lectin leg family contains the legume lectin domain. So it is telling you about the domain, and then it is saying you two things: alignment and envelope region. Okay, and this is a one algorithm. On that basis, it is saying 2 to 238. Okay, this alignment basis, it is saying alignment means in the database, it is aligning with the other proteins and telling That 27 to 258 is a lectin leg B family. On the basis of HMM, it is saying 2 to 38. And on the basis of envelope means the domain envelope. Okay, from where to where. So this domain always is little bit bigger than your alignment. Okay, so it is taking from 26 to 267. So it is always better to consider this one or this one, not this one. Okay. So this way you will get the your protein family. If you want to read more about the protein family, that click on that. Okay, you got now a story here. Like what is the lectin leg B family? In molecular biology, the leguminous lectin family is a family of the lectin proteins. So all these descriptions, like I don't think we need to go. Just uh, for the understanding, yes, this is a member of the leguminous lectin family, which we know from the beginning because this is a known and annotated sequence. But if suppose in another case you got a novel gene, how you will characterize? So for that, this exercise is required. Now, after knowing the family, now we'll go about the structure. What is the structure of this protein? Okay, so if you know in the protein, there is a in the protein hierarchy there is a four type structure. One is the primary structure, second is secondary structure, third is tertiary structure, and fourth is quaternary structure. So primary structure, when we see any sequence of amino acids, that is a primary structure. So you can see here clearly, this is your primary structure. Agree? Now next question comes. So primary structure I know, but if you see in the primary structure in the protein structure hierarchy, there is another form of the structure that called the secondary structure. So how you will get the secondary structure? Anybody can answer how you will get the secondary structure of this amino acid sequence or protein sequence? Okay, so no problem. I will show you how to get the secondary structure of this protein. So for that, you go again in the Google and put the one word that called the PSI spread. I request all of you to please follow me because if you will follow, then you will be able to generate this information. If you will not follow and you will try later, maybe you will not get success. Okay, paste your sequence here. So in this, you take there are several options. One option is the sequence data, like what type data you are having. So we are having basically only the primary sequence or primary structure of the protein. So that means amino acid sequence. So that let's paste that one. Another option is the PDB structure data. So but because we are having only the primary structure, so we cannot put the PDB structure here. Then there is several type analysis this PSI pred perform. One is the PSI pred, that is the predict secondary structure. Another is the DISO pred that tell you the disorder region in the protein. Third one is the membrane helix prediction. Fourth is the threader. So there is several other type like the domain prediction, function prediction. Other servers are also there, but I preferred only to use this PSI pred because if I want to know the name of the protein, if I want to know the secondary structure of protein, this is the most preferred platform. Then let's go to the job name. Just put something like CPL. Okay, chickpea lectin, CPL. Here, sometimes the server is like now it's all together like 16 people is participating and one is me. If 16 people will run together, this server will be slower. Okay, it will take time to give you the input. So we can put some email here. Okay, your personal email ID you can put, and then you can say to program to submit. So now it is failed. You have no authority post. Oh, sorry. Okay, I did one mistake. Sorry. Okay, because it should be in the faster format. So I am not having that greater than symbol and it is CPL. Okay. Okay, let's run now program. Sending job to. Oh my God. You have no authority to post job. Either you are not logged in. You too many. 15 concurrent jobs running. Oh, I hope all of you have started parallelly. Before I clicked, you have started. So then 15 jobs are running. So it is not accepting my job. Have you started the running job?
Sir,
Anybody started running job, sir? Because it is not accepting my job because it is saying all together 15 jobs are running
no,
actually.
the participants cannot disturb our meeting. They can do in their individual devices.
Yeah, they can do only their individual devices, but what will happen because server is same na for this program. So if people will run parallelly same job na, then it will not accept. Okay, no problem. I will show you another server for this purpose. Okay. If it is not working, but this is my favorite one, so that is the reason I was trying to run here. No problem. Okay, so I will run JPRAD actually. PSI Prad is not running. Let's run JPRAD. So this JPRAD is also works for the secondary structure. Okay, so you see here also a secondary structure prediction server, and they have given one sample. So what we can do, we can just paste our sequence here, and then we can say to program make prediction. Now it will take its time to run. Meantime, I will tell you more about the structures of the protein. Okay, so now they are running this job actually. So they are saying this many PDB they found actually. Okay, we are not interested in the PDB structure now. We are interested only in the uh, secondary structure of this protein. Okay, so let's let it continue this one. Okay, it will find its it will take its time. Meantime, I will tell you more about the protein structure. Okay, so why we sorry. Okay, so meantime that job is running, I just want to discuss little bit about the protein structure actually. I told you that protein structure is having the hierarchy like assistant professor, associate professor, professor, and senior professor. Same thing here. Protein structures are also having primary structure, secondary structure, tertiary structure, and quaternary structure. So why we have to know about the protein structure? First question comes, and what is the hierarchy? If you see just now I mentioned when you are talking about the amino acid sequence, that is your primary structure. When you are telling about the secondary structure like alpha helices and beta sheets, that makes your secondary structure. And when these alpha helices and beta sheets folds, okay, they makes the or arrangement of alpha helix and beta sheets, you can call them as a tertiary structure. And when more than one polypeptide chain come together and they form the structure that called the quaternary structure. So why you have to know about the protein structure? If all of you know, in the previous class also I discussed you, like there is a lot of technique has come which, and particularly NGS, next genome sequencing. If you remember earlier days people was performing Sanger sequencing for DNA. And it was taking the months to years to get a one genome sequence. Forget the genome sequence, even like partial genome sequence also it was taking the years. But NGS has made the life very easy. Now you isolate any new organism, Take that organism or take strain. Okay, isolate the genomic DNA, send for the NGS. And after the NGS, you will get the plenty data of the genome of that particular organism. Now, if you want to analyze that genome, you can do by the servers, but that will tell you about only the gene, not about the protein. So if you see in today's date, actually, almost 100 million nucleotide sequences are available from the 300,000 different organisms in the primary nucleotide database. And primary nucleotide database, if all of you know, that is the GenBank, DDBJ, and EMBL. So you see, this much sequences are available, and it is not like that nobody is trying. People is trying because gene is not only enough to tell the function of the that particular gene. Only nucleotide sequence is not enough. So then what you have to do? You have to translate them. So almost now from this much data, people have translated 5 million of these nucleotide sequence and deposited in the UniProt data. That is database for the primary database for the protein. So almost 5 million data sets are activated. So now first thing, you had a lot of gene data that has been translated. Some part of that people consider only 10% has been translated until now. So still 90% is remaining. 5 million is translated, and you can see by your own 10 million means 5 million. So only 5% actually has been translated. Now this 5 million we have to, because okay, I know the protein name or I know annotation. That is not working. I should know the function of the protein also. So to know the function of the protein, it is very important to know the structure of this protein. For that, we are talking about the protein structure hierarchy. Now, how you will determine the protein structure? So primary structure, just you go for the sequencing, you will get the primary structure. Secondary structure, there is a method that called the, there is a technique that called the CD spectroscopy that will tell you how many alpha helix and beta sheets or how many random coils are present in your protein. Okay, but that is possible only after purifying the protein. And today's in the class we are talking about only the in silico methods. In silico method, that is the reason I saw you. One is the PSI PRED and another is the JPRED. So this servers without purifying the protein and using the CD spectroscopy, you can tell how many alpha helix and beta sheets are present in this protein. Another thing is the, so secondary structure you will go by the, you will get by the, if you want to get in silico, you will get by the either JPRED or PSI PRED. If you want to perform the in vitro experiment, then you have to go for CD spectroscopy. But if you want to determine the tertiary or quaternary structure, neither CD nor primary will tell you. So for that, if you want to get the three-dimensional structure of protein, there is a two types of the method. One is the experimental method and second is the computational method. The experimental method is the NMR spectroscopy. NMR means nuclear magnetic resonance. Second method is the cryo-EM, cryo-electron microscopy. And third method is the X-ray crystallography. Similar way, you are having the computational method. So computational method is also, there is three types of the computational method. One is the homology modeling, second is the threading, and third one is the ab initio method. I will just tell you a little bit briefly about these methods. So first thing is the NMR spectroscopy. So NMR spectroscopy is in the field from the several years. Okay, it is in the beginning of the 1900s it was available, but to determine the protein structure, it came in the use in the 1946. Okay, when Bloch and Purcell has used this one to determine the structure of the protein. So NMR, by using NMR spectroscopy, you can determine the molecular weight up to a 50 kDa protein. Okay, but you can get a good resolution for the protein which is having the molecular weight up to 30 kDa. So the most preferred protein is up to 30 kDa. If you want to determine the molecular weight above the 30 kDa, then NMR is not a good option. NMR tell you this, NMR tell you about the distance between two hydrogen atoms. Okay, because NMR is performed in the solution, you purify the protein, and in the solution you take the spectra. Because it performs in the solution, it can tell you the dynamics of the protein. As we all know, the protein is never present in a single conformation. It is always present in a dynamics. It is not like if you are having a protein in a solution, it will be like this. No, it will be always change its conformation. Okay, it will present in dynamics. So the dynamics you can tell by using the NMR spectroscopy. But for the most important thing for this is you should have the soluble and non-aggregating protein. So getting soluble, I have discussed a lot of methods. Another technique which is routinely used to determine the three-dimensional structure, and recently it has received the Nobel Prize in the 2017 for the cryo-electron microscopy, and this is a very recent technique where people have used this technique for the to the determination of the three-dimensional structure of the protein. So this has recently come in the market actually, and that is the reason recently 2017 only they have Dovochet, Frank, and Henderson has received the Nobel Prize for developing cryo-electron microscopy for the high resolution structure determination of biomolecules. So biomolecules, if you remember, I told you it can be a DNA, it can be a RNA, it can be protein also. Okay, but because mostly people, because protein is quite big, so they talk about the protein. Okay. By using electron microscopy, you can determine the or you can obtain a structure with the low to medium resolution. Because this NMR is used in the only solution form. Here, because you are using a one word cryo, so because to keep stable your protein, you add the cryo. So then dynamic information you will get very limited. But in NMR, because we are performing at the solution level, so then you will get a lot of info. But here you will get only limited information. This is good actually for the large molecules and complexes, and it provide you insight about the large molecular assembly. And it can be used for the with the small, very small crystals. And then third crystallography, that is the bread and butter of my my family and my lab people actually, that is the X-ray crystallography. Okay, so X-ray crystallography was first if you all of you know, X-ray came in 1900 when Rontgen has given the when he gave the X-ray. But X-ray diffraction for any protein came in the 1934 when Vernal and Crowfoot Hodgkin produced the first X-ray diffraction pattern of a protein, and that was a pepsin. And pepsin, I hope all of you know, that is this is a digestive enzyme. Okay. So X-ray crystallography is a method to determine the alignment of atom within a crystal. It enables us to visualize the protein structure at the atomic level and enhance our understanding of the protein function. Additionally, we can study how protein is going to interact with the other molecule, how it is going to have the conformational changes from one stage to another stage or after interacting with another molecule, and how it performs catalysis of any particular enzyme. This method is used for the structure and function to determine the structure and function of many biological molecules, not only the protein. It can be used for the vitamin also, it can be used for drug, it can be used for the nucleic acid like the DNA, RNA. So in the X-ray crystallography, like as you see, there is two words. One is the X-ray. The X-ray Rontgen has given. Another is the crystallography. So crystallography words has come from the crystallization. Study of a crystal using X-ray is the X-ray crystallography. So there is three words. One is the X-ray, another is the crystal, and third one is the study. So now X-ray Rontgen has given, that is physics part. The mostly people is here from the biology. We will not put our head there. We will talk only about the second part and third part. Second part is the crystallization. What is the crystallization? So crystallization is a process which is governed by the two factors. One is the thermodynamic and another kinetic, which arrange your molecule in a natural manner to form a repetitive three-dimensional reticula, and that called the crystal. So here I am having a one diagram for you. So you see this is a protein. So you take a, suppose you take a Eppendorf tube. Okay. In the Eppendorf tube, you add your solution, and the solution contains the protein. Now what will happen now when it is present in the solution? You cannot perform the X-ray crystallography, because all of us, I am pretty sure in the in the 30 or 40 years, you definitely got a chance to visit a doctor and go for X-ray. So for, if you remember, in X-ray, you get two types images actually. One is black and another is white. What is black and what is white? Black is your basically background, and white is your bone actually. So you get two types. Okay, so if you want to get for the water, you cannot get X-ray, because you should have a media from through that your X-ray should not able to pass, like what happens in our bones, because X-ray cannot penetrate our bones, so it hits and then diffracts, and that is the reason you get a impression of the bones on the X-ray film. Similar kind here. If you will take in the solution and if you try to hit, it will easily go through this, and then you will not get the diffraction. If you want to get the diffraction, it should be always in a solid form, what we get in a hospital. So similar thing here. So you take a protein molecule, and protein molecule is present in a solution. Now you add a precipitant. What is the role of the precipitant? Because when protein is present in the solution, there is a lot of water molecule is around to the protein. But when you will add any precipitant, those precipitant molecules will start absorbing that water molecule from the solution. And they will start taking away the water molecule, because when protein molecules present in the water, they're easily mobile. Some molecule is here, some molecule is there, some molecule is here. Now when precipitant will start taking away that water molecule, and these protein molecules which were away from each other, they will start coming closer to each other, and that way they will make a reticula like this. See, several protein molecule has come together, and you can see these four molecules have come together, and they formed a crystal. So in the crystallization, there is a two phase actually. One is the nucleation. Nucleation means seeding. Like when you added precipitant, firstly, a small seed will come, means 2 or 3 protein molecules will come together, and around that, then later other molecules will start coming together. So when you are having 1 or 2 molecules, that will call the nuclei. That called and the procedure called nucleation. And when other molecules will start coming closer, to this nuclei, then the size of this nuclei will start growing up. Okay, and that will call the crystal growth. So nuclei generally you cannot see even using microscope, but crystal growth, if the size is enough, then you can see by the microscope. Okay, so what are the important factors which play a role in the protein crystallization? First is the purity of protein. Your protein should be 95% pure actually. Another thing is the protein concentration. Because you see this method you have seen, there you have to bring your molecules together. If you want to bring your molecule together, then there should be enough molecules also. No? So for that you have to look about the protein concentration. The protein concentration should be very high. Generally, in thumb rule in the laboratory, we believe it should be around 10 mg per ml. But then it varies from the varies according to the size of the molecules. Another thing is the precipitating agent. What is the precipitant you are using? So precipitant can be anything. Precipitant, even some protein has been precipitated in the water also. Why is it precipitated? If protein concentration is very high, they will precipitate in the water itself. Now another question will come, what is the difference between the precipitation and crystallization? So just now, if you remember, I gave you the definition of the crystallization. When those protein molecules come together in a natural manner. Natural manner means systematic way. Like one molecule has come here, another molecule, third molecule, fourth molecule. That is the crystallization. Precipitations means they come abruptly together. Okay. And they don't follow, they don't form any pattern, they don't follow any pattern, and they form a precipitate. That is a precipitation. So protein concentration is very important. Neither should be very high. Very high, there itself, this protein makes precipitate by its own without adding any precipitate. Other thing, it should not be very low. Like you are adding precipitate, but still it is not able to remove the water molecule. So that in this case, protein concentration should be high. Okay, around 10 mg per ml. Then another is the precipitant. So precipitant, generally people use precipitant as a salt, like salt as a precipitant. Salt means you can use sodium chloride also as a salt. Ammonium sulfate as a salt, ammonium sulfate as a precipitant. Ethanol also you can as a precipitant. So there is a big range of the precipitant. There is a specific category of precipitant that called PEG, polyethylene glycol. So PEG is also considered a very good precipitant to form the crystal. Another important factor is the temperature. All of you are aware that protein is highly temperature sensitive. So if suppose you are having a protein, you purified protein after a lot of efforts, and after purifying the protein, you have kept protein at the room temperature. Protein will lose its natural conformation, and once it loses conformation, it may first thing it may precipitate. Another thing, if it doesn't precipitate, it may denature also. In all those cases, you have to be very careful with the temperature. So that is the reason generally crystallization is performed less than the ambient temperature. And I'm pretty sure all of you are aware about the ambient temperature. Ambient temperature is the 25 degrees centigrade. So temperature is always preferred a critical factor. Next thing is the pH. What is the role of the pH? pH role is, if you remember when we were performing the probe palm, that time I told you what will be the buffer for this protein. So if you remember the chickpea lectin, the pI is around 5. So what will be the what will be the buffer for this protein? So you should use a buffer plus or minus 1 of the pI of your protein, because at the pI your protein is having the 0 charges. So now you have a buffer either plus or minus. Now you say again confusion has come, why plus or minus? Plus or minus vary actually according to the protein. And we always try to perform any reaction or any activity or anything or any crystallization always near to physiological pH. As all of you know, physiological pH is the 7. So if you want to perform the purification or lysis or any step, you should try to bring your protein near to the physiological pH. Now, if you remember, the pH of this protein was around 5 or 5.5. So what we have to do now? We should go minus or we should go plus. In this case, we have to go plus, not minus. If you go minus, then what will happen? It will go from 5 to 4 actually. That means your protein has gone far away from the physiological pH. Agree? Since it has gone far from physiological pH, it may be highly acidic also. And it may precipitate at that pH. So that is the reason it is not preferred. So what you have to do? Instead of minus, because it is already in minus, now you have to plus actually. So when you will plus, it was already 5. And you will plus 1, it will become the 6. And 6 is more closer to the physiological pH than the pH 5. So that pH is another important factor. Similarly, if suppose pH is the 9, so instead of now in this case we have add up 1. In this case we have to substitute 1, okay, from the value. So if you are having 9, then you use the pH around 8, okay, and there is a different kind of buffers which work for the different pH actually. Another important factor is the additives for the crystallization, protein crystallization. Additives, what you can consider additives? Additives means any additional thing beside the protein and your precipitant will call the additive. Why you need an additive? Now suppose you are having a protein and that you have purified with your lot of courage while it was having the transmembrane region. Now you have purified, and after purification you want to perform the crystallization. If you will not add the detergent, that transmembrane region will interfere in the folding of protein, because transmembrane region is having nature to go and insert in the membrane region. And unfortunately, now we have purified the protein, so it is not having the membrane region there. So then it will create problem in the folding. By nature, it should be trapped this way, is it? But it is not able to trap because there is not any membrane. So then it will, this part which should be trapped in the membrane will create the hurdle in the folding of the protein. So you have to add, then in that case you have to add some kind of detergent. Another, so detergent will keep this end free, so then they will not able to interfere. Another is the reducing agent. If you remember when we were discussing over the protoparum, we have checked in the one most important thing is the cysteine actually. How many cysteine is there? Like suppose your protein is having 6 cysteine. It may, theoretically it may form minimum 3 disulfide bases. So those disulfide bases will change the, like suppose your protein should be present in this form. Now with this disulfide bridge will bring this form together. Okay. So then this will change the conformation of protein. To avoid this oxidation of cysteine, we add some reducing agent. Generally people use beta-mercaptan or DTT, dithiothiol. Sometimes what happens, if your protein, like just now I mentioned, your protein is present only in the dynamics. And crystallization you are performing in the solution. So it will be always like this way, this way, this way, this way, this way. Then those molecules will not able to come together in a proper format, in a proper form. So that means to stop these dynamics, because they are always in this. What we can do, we can add some substrate or cofactor. So what does substrate and cofactor do? Because most dynamic part of the protein is always its catalytic side actually. So if you will put your substrate or cofactor, because those additives will go and bound with that region, active site. If they will bind to the active site region, then it will lose its dynamics. Okay, it will stuck again in a one form, and that you need for the crystallization. Even like I am having on my example, like when I was working in Singapore, I was working on a protein that called FKBD. FKBD is the FK506 binding domain from the plasmodium. It is a validated target, like you can use this FKBD as a drug target against the plasmodium. When I tried to crystallize my protein with the FKBD, I tried 1 year, I didn't get any success. Then I discussed with my mentor, and then we realized, oh, maybe this protein is highly dynamic actually, or it is highly flexible. Let's try to perform this with the its cofactor or its substrate. So substrate was the FK506. And we added FK506, protein crystallized happily, and we got the structure. And since we got the structure, I got a position. So additives are very important for the crystallization. So how you will perform the crystallization? So for crystallization, there is several method actually. Very common method is the vapor diffusion. Another is the batch crystallization. Third one is the microdialysis. Like what we do in the dialysis, exoosmosis, endoosmosis. Similar kind you can do in the microdialysis. Liquid-liquid free interface diffusion, seeding. So among these actually, vapor diffusion is the most commonly used method. So I will talk about the other, only vapor diffusion method. Other methods like batch methods, microdialysis. Microdialysis is very simple, like just to take your protein in a, like what we do in the, like take a buffer, and in that you take, because you cannot take a membrane, dialysis membrane, bigger membrane, take this small membrane, put your protein and put in a precipitant. The container should contain the precipitant instead of any other buffer. So then what will happen, because protein is present inside and precipitant is outside, then exchange will start happening through the dialysis membrane. And at one stage it will form the crystallization. Okay. So these are the different methods. So let's go, like most common method is the vapor diffusion method. Vapor diffusion method is also having two types. One is the hanging drop and another is sitting drop. So if you see here in the hanging drop, in hanging drop, you take a like 24 well plate. In that you are having a one bell, suppose. In this bell, of the top, you put a grease actually of the outside of the this bell. So this is a, like you can see here, this is your grease. Okay, this, and after putting grease, you add your precipitant here. So precipitant, suppose like 4 molar NaCl I have kept here. Now you take the cover slip to cover this bell. On the cover slip, you put your protein, suppose 1 microliter protein, and take 1 microliter of this reservoir and mix with the protein. Now suppose you have taken in reservoir, suppose 4 molar NaCl. What will be the concentration of the NaCl in this drop? It will be the half concentration from the reservoir solution. Suppose you are having 4, so then here you will have the 2 molar NaCl concentration. So 2 molar NaCl concentration is there and 4 molar is here. Now you take this cover slip and put on your bell. Then you will put on your bell and then close this and then leave it. Now what will happen, because on top NaCl concentration is less and bottom NaCl concentration is high. So then water will start coming down actually from top to, okay, it will start evaporation actually. So now when it will evaporate, water will go down, but NaCl, which is having high concentration in bottom, that will go up. So then the concentration of NaCl will start changing here, and protein molecule, because NaCl, so water is going, NaCl is coming, so water is going. So what is happening now? The concentration of protein, suppose it was 10 mg per ml. So in the drop, it will be 5 mg per ml because you diluted. Now when water will come out and NaCl will go there, So then concentration of protein will start going changing. It will 5 mg per ml to become the 6 mg, 7 mg, like something. Okay, nobody knows exactly what is the concentration, but there will be some definitely exchange of the water. Okay, when this water exchange will happen, the salt concentration has changed and salt concentration will protein concentration has changed and protein concentration change will bring your protein molecules together. And when they will come together, they may form the crystal. Okay, so the similar way, there is another type of the vapor diffusion is the, because here you are diffusing the vapor. So similar kind, there is another method that's called the sitting drop method. Instead of hanging your drop on the cover slip, now you get a stand. On that you put your protein and precipitant together and put cover slip on top. Okay, so cover slip will be on top, so it will create a vacuum. So nothing will able to come from outside and nothing will able to go outside from this bell. Okay, so this is the principle. If you see, both are similar methods. They are identical. Only their mode of the setting the drop is different. In this you are hanging. In this you have kept on a stand. So when you will perform a crystallization, you will get this type crystals. So these are considered very good crystals, and I hope all of you have seen sometimes this type crystals in the books and somewhere else. Okay, these are the single crystals you see. Okay, but here you can see several crystals are there. Here two crystals are there. So it varies actually. Sometimes you may get one crystal or you may get the 500 crystal also in a one bell. And this crystals you can watch only by the, you can watch under only the microscope. So, so this type crystals you will get. Now you will ask another question actually. As we know, all of us know that crystals are, there is another crystal also that's called the NaCl crystal, salt crystal also. And that is available easily in market. So then why we are behind this crystal actually? So there is some difference between the protein crystal and salt crystal or mineral crystal. Protein crystal, because if you remember, you are removing water when you are performing the crystallization, but you cannot remove completely. This water molecule will also pack along with the protein molecule when it is forming the crystal. So that is the reason you will have the very high solvent content. Solvent is here basically water. Okay, so you will have almost 30 to 80% solvent content. But if you remember in the market, you have seen salt, NaCl crystal. If you break them, there will not any water molecule. So generally they content either zero or very less solvent content. Protein crystal is very soft and crushed easily. They're very delicate, highly delicate. But salt crystal, like if you want to break a NaCl crystal, you have to apply a lot of force, then only you can break. So they are very hard. Since they are soft and crush easily, and they are having a lot of water molecule or solvent content, they are sensitive to dehydration and change in the pH or ionic strength. But mineral crystals, because they don't have the solvent, they are very hard. They are resistant to anything. Like if you buy any time this salt crystal from market and put even in at any temperature, if you will go very high temperature, only they may melt actually. Before that, they will not be melted. If you want to get a protein crystal, for protein crystal, nothing is fixed actually. What is going to work as a precipitant, nobody knows in the world actually. Even the people who has determined 100 structure, and even recently 2010, when Indian got a Nobel Prize actually, Venki Ramakrishnan, to determine the structure of ribosome, bacterial ribosome. He got the Nobel Prize. He can also not tell. Okay. So crystallization conditions are not predictable. You have to try 100 conditions. In my lab, in our lab, we try almost 1000 conditions in one attempt to get a protein crystal. But if you want to get the salt crystal, what will you do? You just change some temperature or you remove some water molecule, they will form happily this salt crystal. So this is the basically difference between the protein crystal and salt crystal. Now see, I am talking about the crystallization. Now let's come back actually. I told you three things are there. One is the X-ray, second is the crystal, and third one is the X-ray. Now you are having X-ray also, you are having the crystal also. But how you will determine? Suppose X-ray is here, it is available in any hospital. Okay, I will go tomorrow protein, I will go protein crystal and I will take hospital. Whether they can determine structure? No, they will not able to determine structure. For this, you should have the particular optimum beam actually. Beam size is also defined. But this is very small protein crystal because whatever we are facing in X-ray, that beam size is very big actually. Okay, here protein crystals are only visible, they are not visible by the naked eye also. They are visible only under the microscope. So for that, how you will get the, how you will determine the structure? You have grown the structure, you know you are having X-ray, now how you will determine. So this is just brief actually. So suppose you have kept a vapor diffusion by the hanging drop method. You got this crystal. Now you take your crystal to the X-ray beam. An X-ray beam should be on the 1.5 angstrom. Beam size should be very small. Okay. And now suppose this is your X-ray beam. This is your crystal. You put crystal just in front of the X-ray beam. So what will happen when this X-ray beam will hit the crystal? It will diffract. So this way you will get a kind this diffraction pattern. There you will get a several dots actually. So these dots indicate the presence of the molecules in the crystal structure. Because crystal, sorry, in the crystal, because crystal is made by the protein molecules. So those molecules arrangement you can see here. But you cannot see this is my A molecule, this is my B molecule. That you cannot see. Only you can say yes, you got some pattern using diffraction. When you perform diffraction, you should remember one more thing. What happens if you remember, like when we visit X-ray for X-ray, always doctor asks us, have you gone any time in the last 6 months for X-ray? If you say yes, then doctor say no, you cannot go X-ray for now. Wait for some time. If you say no, I have not gone for any X-ray in recent times, you say okay, now you go for X-ray. Because what happens, X-ray is ionizing radiation actually. And ionizing radiation will affect, ionize the media. So this will go through that, it will pass. So to avoid that ionization, doctor says always do it after 6 months for the X-ray. Similar way in the crystal, what happens when this X-ray will hit the crystal? There will be some diffraction, but that heat which has been generated, that may kill your crystal because your crystal is having very high water molecule, and if you are having very high water molecule, it may melt very quickly. To avoid that thing, what you have to do, you have to freeze your crystal. How you can do? How you can freeze? Suppose this is your crystal, this is your X-ray beam. From top, you start pouring the liquid nitrogen. When you will pour the liquid nitrogen off, not X-ray beam, you have to pour on your crystal. So all of us know X-ray. What is the liquid nitrogen temperature? Okay, nobody is interested to tell the liquid nitrogen temperature. Liquid nitrogen temperature is minus 196 degrees centigrade. So 196 degrees centigrade, when you will pour the liquid nitrogen of the 196 degrees centigrade, it will freeze your crystal. Okay, and the heat which is generated by the X-ray, that will be nullified. So that comes under the cryocrystallography. So anyway, this is your crystal. Okay, and from top you are pouring the liquid nitrogen, and after some time you got this type pattern. So only one pattern is not enough. You have to generate several pattern of this. After generating these patterns will not tell you what is the structure of the protein. Then you have to take these patterns and process them and find a phase information using a computer. Because computer is having its software. You download the software like a CCP4 or Phoenix on your computer. Process this data there, and after processing will have the integration, sorry, indexing, integration, merging, and then scaling. After scaling, next step will be the phase information. Phase information means this is telling the density of the individual amino acids, but how those will fit with each other, that you will get from the phase information. For phase information, you can use the molecular replacement, most commonly method, and other methods like Matt said. So I don't want to go in the detail. It is not helpful for you. So just I want to tell you how to determine structure. So the computer now, because this is completely experimental from here to here, it is completely experimental, which you can do only in the laboratory. But when you sit on the computer, then you can process the data using the different softwares like the Phoenix or CCP4, and at the end you will get a structure like this, whatever you are able to see here. Okay, so this is about the crystallography. Why I am emphasizing on the crystallography? Why it is considered the best method to determine the three-dimensional structure? If you see in the history to till now, almost 29 prizes for 48 laureates had been given Nobel Prizes for the crystallography. And that is the largest number of Nobel Prizes which has given to any type science. And that goes for the crystallography. 29 prizes for 48 laureates. This started because with the search of the X-ray in the 1901, with the Nobel Prize for Physics awarded to the Rontgen for his discovery of the X-ray. And recently in the 2013, Karplus, Levitt and Barsal, they are the latest one who got the Nobel Prize for the X-ray crystallography. They have received the Nobel Prize for the development of multi-scale models for the complex chemical system. So this is about the crystallography. Now, when I was discussing about the database, primary database. So for protein, I told you some database like a UniProt and PDB and per PSD. So these databases are primary database for protein. That time I told you about the PDB, protein database. The protein data bank is a place where people submit their protein structure along with their protein sequences. That is the reason it called the primary database. But this database is only for the three-dimensional structure. If you say, oh, I have determined the primary sequence or primary structure of a protein I want to submit, you cannot submit there. You have to go for the UniProt, or you have to go in the NCBI and submit there. You cannot submit here. Here only you can submit along with the you can submit only the three-dimensional structure along with the protein sequence. That is a complementary. Like when you go a restaurant, you take a dessert. It is like that. It is not a main stuff because nobody goes in the restaurant for the dessert. People visit actually for the main stuff. So this PDB database is actually for the three-dimensional structure of the biological macromolecules. So biological macromolecules, anything actually, it is a protein, it is a DNA, or it is a RNA. And this data is available for everybody. Just now I will give you one demo also for this. Just I want to discuss some theory, then I wanted to go for the demo. That is the reason I just showed. All data are available to public. Anybody can extract any data from the PDB database, like whatever you are extracting from GeneBank or UniProt. Similarly, you can get a data from the either you want any structure or it is completely free. No, you need not to pay charges. Okay. If you see in the protein database, if you consider if 100% structures like 100 structures are available, among them 88% structures obtained through the X-ray crystallography, while only 8% by the NMR and only 4% TAE. So you can see the difference, what I was telling in the beginning, because why people is getting less structure by NMR, because NMR is not good for the big molecule. Like just now I mentioned, Venki Ramakrishnan got the Nobel Prize for structure of ribosome. Ribosome size, if you see, it is in the more than 500 kD because it is having seven polypeptide, it is having RNA molecule, a lot of things are there. Okay, so can you determine the structure by the NMR? No, you cannot determine ribosome structure by NMR. You can determine only structure up to 30 kD. But if you want to determine above 30 kD, then what is the option? Earlier option was the only X-ray crystallography. From 2017, just 3-4 years back, this cryo-EM has come in the picture. Okay, that is the reason just 3-4 years, so it is having only contribution only 4%. But yes, this percentage will increase because this is good for the big molecules or big assemblies like ATPase, ribosome, these big molecules, PKS, all these big assemblies, it is good actually. But since it came recently, that is the reason Venki Ramakrishnan has spent 10 years of his life to determine the ribosome structure. He started his research in 2000 and he got a structure in 2010. So 10 years he spent to determine the structure by the X-ray crystallography. NMR was no choice. Only choice at that time was the X-ray crystallography. Cryo-EM was not there that time. It just came in the last of the 2017. It came in the market. So that is the reason. If you see, and another good thing with the X-ray crystallography is the range actually. If you want to determine structure of the ribosome, which is very big molecule, you can determine by the X-ray crystallography. And if you want to determine structure of a small peptide also, you can determine by the X-ray crystallography. So that range is very vast for the X-ray crystallography. While NMR is having that limited range. It is good for the small molecule. So this PDV data bank was established in 1971 in the Brookhaven National Laboratory in New York. Okay, this is synchrotron facility where they have established. And in beginning, they were having only 8 structures in the 1971. And later this, because this is a basically a X-ray facility where people go and collect the data. Okay, I have collected in 2015 here. So then they realized because managing them is difficult. So then they have made a consortium type things that called the RCSB, Research Collaboratory for Structural Bioinformatics in 1998. And now that RCSB is maintaining this protein data bank here. So if you see, I told you in 1971, they were having only the 8 structures. But if you see today, this is yesterday night for your information, I have collected the data statistics. It is 1,75,759 structures are available there. So you can see like how many fast it is working. So this is the protein database. This is the protein data bank homepage actually. So this one I will show you when after giving my class actually I will give you the demo of this, how to use this PDV site. So you can see http www.rcsp.org. Okay, this is the recent holding of the PDV. Yesterday night around 12 o'clock I have made this table for all of you. So if you see here, like X-ray, NMR and EM. And if you see here, total number. So total number is 1,75,759. If you see, in the case of protein, almost 1,53,000 structures is having the protein. So you can see almost 90% structures belongs from the protein. And see another thing, out of 1,75,000, you are having 1,54,000 structure determined by the X-ray. Only 13,000 structure by NMR and only 7,000 structure by EM. People have clubbed also 2 or 3 techniques actually, and that technique called hybrid actually, multiple methods. So that is also very less. So people generally most preferred method is the X-ray, and most preferred biomolecule is the protein. People have determined protein structure with the oligosaccharide, like any carbohydrate body. Okay, with the nucleic acid also. Okay, almost 9,000 structure with the oligosaccharide and nucleic acid is available. Only nucleic acid also they have determined actually structure like aptamer. Okay, aptamer also they have determined structure almost 3,500 and others. Okay, so this way if you see 71 and today is the 21. So only 50 years. The number of you multiply, I don't know how many times actually, has reached from 8 to 1,75,000. Okay, so this is the PDB. Now, because you can say, sir, you are confusing us, you are showing three methods to determine the experimental structure. So, which one you have to use actually among these? So, that depends actually what is available with you. Like in University of Hyderabad, we don't have diffractometer, X-ray diffractometer. But I have X-ray crystallography, so I have to use the no other choice. I have to use the for that either I go to IICT or I go to CCMB or I go to ISC Bangalore. But for me, that is my bread and butter, so I have to go for that. But for a new person who wants to just determine a structure, for him it doesn't matter. It is X-ray or NMR. So then you have to look what is the available in your institute. Okay, or available in your periphery, or whatever is accessible to you. So that is completely your call. Second thing, yes, important thing is the size actually. Like suppose if you are having a protein of 100 kb, then you cannot definitely go for NMR. Another thing is the cryo. So in India, cryo we are having only one or two places. So only the option is the X-ray. Like in Hyderabad, we are having three diffractometer. That is the reason funding agencies are not ready to give to University of Hyderabad. They are saying already CDFD, CCMB, and IICT is having. Why University of Hyderabad needs? Let's use with them. You know, funding agency is giving. But yeah, in Hyderabad you are having that option. But cryo only NCBS Bangalore is having. Nobody else is having. So availability of technique, one important thing. Second is the well structured protein, easy to grow crystals. If your protein is properly folded, you can grow easily crystal. If your protein is not easily folded, then you will struggle. So better you go in that case, you go for NMR. Don't try or spend your time for the X-ray. Largely folded protein, anyway, you have to go for NMR. Now, another important thing is the dynamics. If you remember, when I was telling about the brief about these three techniques, there was one more important point actually. Like in the crystallography, you are pouring the cryo liquid nitrogen continuously, which is having temperature minus 196 degrees centigrade. So in that, all molecules and crystallization, because molecules are fixed in a proper manner. That is the reason it forms the crystal. So then, because they are fixed with each other, there is not any space for them to move. Now they're fixed there, so then you cannot get the dynamic information. If you want to get dynamic information, the best option is the NMR because that you are performing in the solution. Another is information is the if you want to get structure activity relationship from the protein structure, then yes, because there are also dynamics important because activity, how is the catalytic site is moving, how ligand will bind, is easily you can interpret in NMR than other methods. Molecules, if you're having large molecules, then you have to go X-ray because this whatever I am it is in this perspective. Yes, like if you are in US, then like there is several places they are having the EM electron microscope. But here we are having only one place. So fast structure determination, it is generally people consider X-ray, but it depends completely on the your crystal. If you got crystal, you can determine the X-ray structure. But if you don't get crystal, it may take the years. Like just now I gave you an example. Venki Ramakrishnan took 10 years to determine the ribosome structure. So you cannot say this is a fast way. So then that depends completely on the what is the protein. Okay, another is the quick bonding assay. If you want to see the two molecules interaction, you can perform the NMR. Okay, so this is up to the experimental method. Now I will move. Do you want to take any break or I can continue? So do you want to take any break or I can continue?
Sir,
Sir, do you want to take any break for drink or I can
no,
continue?
we are near to the end of the session. We
Oh,
can
is it?
sum up.
Oh, then I have to quick little bit. Is it?
Yeah.
Okay. Okay. So then now next is the because so just now I was talking about the experimental method. Then other method is the computational method, which I am going to give you demo actually. So what is the computational method? Why we need the computational method? Because whatever is the experimental methods are available, they are having limitation. Like X-ray is having limitation. Without crystal, you cannot do anything. Okay. NMR is having limitation. With protein, you cannot do anything. So cryo-EM is not available in India. So limitation. Lot of limitations are available. Another thing, all techniques are very expensive. You have to input lot. You have to spend a lot of money. Then only you can have a setup. So one thing is the problem is the limitation of the current experimental techniques. And another thing is the because just now, if you remember in the beginning, I told you out of 100 million, we are having information of the only 5 million protein. We are having only 5 million protein is available only translation. So now you can understand for the if you want to know about the function, how much. If 5% is there, you consider again another 5% of this. So then we are having just for the some 10,000 or 20,000 protein, which is very well characterized. So if you want to fill this gap from 100 million to this 1,000, that time you can use the computational methods. Another is the protein engineering to alter the function of the protein. Now suppose I am having a protein which is having specificity for substrate A or substrate B. I want to change the substrate specificity with the substrate 3. Instead of substrate B, I want to make some mutation. So for mutation, I should know what is the atomic arrangement of those protein, those residues. If I know atomic arrangement of those residues, I can easily perform the iteration of the function. And very most important thing, the computational techniques used, that is the rational drug design. Okay, so drug designing, that is a very commonly used actually. So there is again another, I told you three methods. One is homology modeling, threading, and elimination. So homology modeling is the simplest reliable approach. Proteins with similar sequence tend to fold into the similar structure. Has been observed that even proteins with 30% sequence identity fold into the similar structure. Does not work for the remote homologs, that is the 30% pairwise identity. Okay, so just I will tell you briefly how to perform the homology modeling and then I will give you a demo also. So now you see, you take a protein molecule, just now we are having this 268 amino acids. Take and then perform the sequence alignment. Why sequence alignment? Perform the sequence alignment where we, if you remember, we were performing the sequence alignment with the database, total database. Here we have to perform the sequence alignment with the structures because we want to determine the structure. So if you get the sequence information, that is not going to give you the structure. So if you want to determine the three-dimensional structure of any protein, you take the protein sequence and look in the PDB database. PDB data is protein database. And there you find a homologs of this protein. Okay, when you find a homolog of this protein, which is having high homology or high similarity or high identity, okay, select the template and then align those two sequences. Okay, because if you remember, like I told you, there is two types. One is the global and another is the local. Okay, so then first thing you have to find a template. After finding, yes, this is having good homology, then you just take extract only that template and perform the alignment. And after aligning, then you go for building a model. Building a model, there is a two way. One is the online way, that is I will give you demos this model, and another is the offline mode, actually that is a modeler. This both method, both will give you the structure. They will build a model of the protein which you have taken as a target. So if you remember, you are having 268 amino acids. So now you will get a model for that 268 amino acids. After getting 268 amino acids, next thing you have to validate that structure. For structure validation, very important factor is the Ramachandran plot. I hope all of you are aware about the Ramachandran plot. It gives information about the torsion angles of the side chain. So because amino acids, if you see the main chain, they are linear basically. But the side chain is highly flexible. Okay. This side chain, how side chain is present in a model, on this basis you have to evaluate your model. If your side chain is perfectly fitting, then you need not to worry. Okay, you are not having any outlier. How you will get outlier at the end, I will show you this. Okay, you need not to worry. But if you are having any outlier, that means your model is not good actually. That means if your structure is not following the Ramachandran plot, you may have some outliers and outliers are not good. So then what do you have to do? This structure or this model, whatever you obtain from this template, is not good. Then you go back and look for the another template. Until you get proper validated structure, you have to perform this methods continuously until you get a proper model. And when you get a model, you go for the analysis further. So you see here, this is your target sequence. You found a template structure which is having sequence homology with this. Then you have aligned, and after alignment, you got a target model, and then you check the Ramachandran plot, and you are getting all residues are in the allowed region. Now, next thing after this homology modeling, another thing is the threading. So when threading, homology modeling is the most common method. It is good actually if you are having the homology, sequence similarity around 30%. But if you are having sequence similarity less than 30%, then what you will do? Will you not determine the structure? Yes, you will determine the structure. We will look for the another method. Okay, that another method called the threading. So threading is a method actually when in the protein, in the homology modeling, you look always for the sequence identity. Here you look for the structure alignment. Okay, so because what happens when any protein makes a fold actually, so it adopts the similar folds despite having no significant sequence or functional similarity. So those folds, because protein structure is made by the folds, so you take those folds, means building blocks. You take those building blocks and then determine the structure. That's called the threading. So comparing parts of the sequence to a database of non-thread amino structures using a scoring function. Okay, this works at least somehow what approximately 80% of new protein, not all protein. So how you will perform the threading? Like suppose this is a protein sequence. Okay, like we are having 268 amino acids. They are not having similarity with any other structure, other structure available in database. So we are going for the threading, and threading can be considered kind of you are looking exploring the protein fold. Okay, so you are having a library of protein fold, like you don't or not, I don't. There is server which are having the library of the different folds on the basis of their sequences. Okay, so this structural folds. Now what will happen? The server will take your protein sequence and will try to fit this sequence in the different folds actually. Okay, 1, 2, 3. Maybe it will firstly fit with this, fit this way. It may not fit maybe your protein sequence. Then it will try to fit this. It may not fit maybe, but it may fit in this. So then it will give you a structure. So this structure will be highly defined, where it got a fold actually. Another structure also it will try to define for you secondary structure, but in some cases it may define good, it may not define good also, but overall structure you will get by the threading. Then there is another method actually that called ab initio method. Ab initio method is not used regularly in the lab because it is having limitation. One limitation is it is not good for more than 100 amino acids. If you see any protein, like mostly proteins are having the uh, amino acids more than 100 only. So then one thing is the problem. And second thing is this is completely prediction based because this is structural prediction. This is actually some algorithm based of this method. So that is the reason it is not in the use actually in the field recently. This is just only for the knowledge. Yes, there is third method also, and only at the end when people don't get success with the homology modeling or threading, then they go for the ab initio. But it is not, reliability is always questionable. Okay, then so now let's come to the what are the programs which can help you to determine the three-dimensional structure, uh, predict the three-dimensional structure, not determine, prediction. So there are several programs which is available like Swiss model, which I will show you now. Okay, Rosetta, PSI Pred, PSI Pred, all of you have seen. That is not working actually. I don't know why. There is a lot of files have been submitted, and then after this you view the your structure uh, that I will show in the next class using the RAS mode or Pi mode. Okay, so then let's see first program this model, which I will give you demo now itself. Okay, so how it works that I will show in the program. Okay, then another program is the Lomets. This is good for the this model is good for the this purpose homology modeling, while Lomets is good for the threading purpose. Okay, and all these are free, so everybody is allowed to use. It is not restricted. It is for central university or state university or institute or only foreigners can use. Okay, now either you perform an experimental method or you perform a computational method. Most important thing is the structural validation. So structural validation you can get by the Ramachandran plot, geometry and stereochemistry of the residue. R factor and CR factor, that is applicable only for the, this called refined factor actually in the X-ray. Correlation between the experimental data and structure. This is very important thing. Like suppose you determine a structure by the suppose X-ray crystallography, and now you see with the your lab experiment like function. So like I will give you my own example like protein which I was mentioning you, which has determined by I tried to determine with the apoprotein. I never got a structure. Then I have added the cofactor and then we got the structure. But when you got cofactor structure along with the cofactor, we were finding a dimer of the two molecules. But when I was performing gel filtration of that protein, it was always coming as a monomer. So that is the reason it is very important to correlate between the experimental data and structure. It is not like, okay, I got structure only, this is correct, or my experimental data is correct. In the science, nothing is wrong and nothing is absolutely correct. A can be correct also, B can be also correct. So this is completely, always you have to correct. You have to correlate. Okay. Resolution of the data upon which the structure is based, that is also for crystallography. Okay. So Ramachandran plot, like I just told you, like it works for the diagonal angles of an amino acid. Okay, so you will have the different type outliers and other things actually. And this is not applicable only for the one amino acid. Basically, it is not applicable for two, but one amino acid because that is not having side chain. That is the glycine. And this is also not applicable for the proline actually because that is also not having any side chain. Okay. So when you will see a standard Ramachandran plot without anything, there is a different regions like beta sheets, right-handed alpha helix, left-handed alpha helix. So some residues will go in this region, some will go in this region, some will go in this region, and some residues will be in the outliers. Like they will neither will go in this region nor this region, so they will come in the outliers. So you see here there is not any outlier. Mostly residues are in the allowed region. Okay, so these are the few softwares which people use to check perform the validation. Okay, so this is the just some basic actually, and just I want to tell you about what we are doing in our lab actually. we are using different aspects of the protein structure actually, three-dimensional structure of protein. And we are using three-dimensional structure for drug discovery. And recently we have published European Journal of Medical Chemistry and International Journal of Biological Macromolecules and CVDD on the drug discovery using the three-dimensional structure. We have given the catalytic mechanism using the three-dimensional structure of protein, and we have published several papers like International Journal of Biological Macromolecules, DVRC, DVA Journal Subject, and we are using this three-dimensional structure of the protein to design the vaccine also. And recently we have uh, designed some COVID vaccines also using this, and that has been published in the Journal of Biological Journal of Biological Macromolecule Structure and Dynamics and International Journal of Applied Research and Therapeutics. So with this, now I will come back to again the demonstration, what I was showing you earlier. Okay, so let's see what happened. Yes, you got the secondary structure of your protein. So, so you see here it is a your, you can see in the PDF also this one. Okay, how is the our secondary structure? Let me open the. So before going for the class, I just tell you about some the JPRAD actually. Now, meantime, this is opening. Okay, this has opened actually, not looking good. Okay, so let's see here, sir. Okay, so this red color you can see here. This red color is indicating your alpha helices, and this green color is indicating your beta sheets. Okay, so what it has done, it has taken from the several available algorithm, and then it has determined the structure. But you see, mostly algorithms are following the similar kind pattern, like they are saying, yes, there is alpha helix in here actually in this region. You can say from beginning actually, and then it is having mostly the that green color. Green color is indicating your beta sheets. Okay, and along with that, you can see some kind of numbers here. That number is indicating the confidence of this program. Okay, so like you see, 9 means very high confidence. Like whatever they have predicted, they are considering, because this is also they have done on the basis of the sequence identity in the database. So they are also running there. So if they are having this sequence there, so then they will show the very high confidence level. And if they are not having that sequence there in the database, then they may show you the zero. Like you can see zero here. That means it is indicating means the confidence level is very low. Means they are saying here you are getting like where let's see, they are not able to find the alpha helix or beta sheet. What they do, these programs put the random coil. So they have kept one random coil here. Similar kind zero is here, so then they have kept a random coil. So wherever they are not finding, they will put a random coil. Here they have not kept random coil because this they are having with the different algorithm. They are finding yes in this region from here to here only except one residue, it is forming a beta sheet. So if you see throughout this, there is a several this thing which is forming the secondary structure. So you can interpret by this way that your protein is very well folded. It is not having any problem in the folding actually. So all of you are able to see the data now.
Sir, there was a question from a participant in the chat.
Yes.
She was asking one question.
Yeah, please.
Regarding in primer designing, we can add poly tails.
Yes.
Instead of restriction sequence. I got that much question in the chat box.
Yeah, actually, yes, you can do anything because that is the molecular biology. Like because you have to add just a adapter in your primer. That's all. Okay. So here instead of restriction sites, because if you see restriction sites is having 6 amino acids, 6 base pair and 3 additional. So altogether 9 you have added. But those 9 should be part of your primer. So because you have added 9, so those will, when your gene of interest will amplify, along with that they will amplify. But now question comes actually why we are having the restriction sites. Restriction sites we have added because we want to make a overhang to put this insert in a vector. But suppose if you add a polyethyl, what is you are going to do? So application, on the basis of any application you can do. If you are having any application where you need a polyethyl, yes, you can do. But that polyethyl will not help you to clone in the gene. For that, anyway, after restriction, after polyethyl, you have to put again the restriction site to insert this gene of interest in the your cloning vector, in the expression vector.
Thank
Okay,
you,
sir.
sir, for your continuous explanation, continuous lecture taking. You have taken more than 2.5 hours continuously your explanation. This makes us to understand that how keen you are about what is say job work given to assigned to you. So it is very fine to see that modern sciences are making a really a great achievements in the field. So we have one more session with you. So hope to see you back again in the next session. So with your kind permission, I would like to ask the participants if they can post something in chat. We will have the interaction with sir.
Yeah,
Please,
please.
please, if you are posting some question, it is
Hey, sorry, actually, I just want to mention one thing that we all are colleague actually. Like, I'm doing job here, you are doing job somewhere else actually, but we are colleagues. So, and science, I believe actually is the my background is not a physics, but I am performing the X-ray crystallography because things comes with the discussion actually. So if you will not discuss and if you will not have query, if you will not think then about this thing, whatever I am telling, then there will be difficult actually to interact. So it is my request actually, please think and if you are not finding any question today, please ask me in the next session. But please discuss. Discussion
how in silico could be a rationale for in vivo. Whether all the whatever findings in silico you find,
Hmm.
they be applicable in vivo means they are uh, appropriate actually. How much?
Yeah, sir. Yeah, if you sir, just I will tell you one thing. Like if you see in the database, we are having only 1 lakh structure. But if you see, we are having the 100 million gene sequence available now. So now one thing is you see how much discrepancies is there actually. It is not even the 1% structures we are having now. One thing. Now and another thing is actually if you are having a high sequence homology or like whatever I am showing in silico, this in silico is based basically whatever the computational whatever information experimentally people have collected now, that experimental information people has kept in a database, and from database people is extracting, and then on that basis they are giving the this inference which we are getting as an in silico information. So this is, we cannot say this is completely unreliable. It is also quite reliable. And that is the reason now recently one more science has come actually, and now even in our university also we are having MSc program on that system biology. So like earlier people was talking about the biotechnology, botany, zoology, everything they were talking. And even they were talking separately computational biology. Now the system biology has brought them together. So firstly, get the information, whatever computational information is available, and then you validate them. So this is all together is making now a story. Yes, whatever is your question, that is also correct. Like in all cases, it is not reliable. That yes, 100% reliability is not there, but you can say 70 to 80%, whatever data people is getting in silico data, that is reliable if you want to perform the in vivo or in vitro study with those reliability, that data actually.
Thank you, sir, for wonderful explanation for the query. With your kind permission, I would like to end the meeting now for the day.
Okay, sir. Thank you, sir.
Thank you.
Yeah.
Thank you, participants. Tomorrow is Sunday, and tomorrow also we have again the sessions. Kindly join us tomorrow again back. Thank you very much for being with us.
Thank you. Thanks to all of you.